Welcome to the Talking Precision Medicine podcast, brought to you by Genialis. I'm your host, Raphael Rosengarten, CEO and co-founder of Genialis, the RNA biomarker company. On this podcast, we discuss the future of healthcare and health technology, and how advances in data and data science are fueling the next industrial revolution. If you have not already, please subscribe to the show on Spotify, Apple Podcasts, or wherever you listen to podcasts. And for more information on Genialis and our unique approach to AI-driven precision medicine, please visit our website at www.genialis.com. That's www.genialis.com. Now, on to the show. What a pleasure to sit down with my longtime colleague and friend, Art Krieg, to discuss his latest venture, Zola Therapeutics, and his adventures in the great outdoors. Art has launched and exited a number of innovative biotechs, each time delving deeper into mechanisms by which we can harness the immune system for therapeutic purposes. Join us as we discuss how, despite a well-earned retirement, the siren song that led to Zola was strong enough to get Art back in the game. Well, it's my pleasure today to sit here with an old friend and colleague, Art Krieg. Art is a serial entrepreneur in the biotech space and just recently announced he's coming out of retirement early with a new project. I kind of knew he couldn't sit still, but Art, your new company is called Zola, and I'm going to let you tell us about Zola Therapeutics. You're going to do a much better job. What is Zola? What's your mission, and what are you building?
Yeah, thanks. What Zola is a new biotech trying to do cancer immunotherapy by activating the immune system. And what we're building is a platform for activating the immune system with a new class of compounds that we think has such broad potential for cancer immunotherapy that we don't want to follow the traditional model of just selling ourselves to a single company. We actually want to structure the company with multiple different immune activators in different subsidiaries, like special purpose vehicles that will be licensed to different pharmas in order to ensure the broadest development of this.
So there's a lot there to unpack. There's a new science, right? So a new kind of molecules and maybe therapeutic modality, but also a new kind of, a different kind of company. And let's start with the science. Tell us a little bit more about how this works. And is it molecular therapy? Is it a cellular therapy? What are the actual medicines here?
The way that this works actually goes back to a concept called nucleic acid immunity, which is the concept that the immune system evolved various defenses to protect our genome integrity. And this started in bacteria with the evolution of CRISPR-Cas systems, for example, to protect bacteria against bacteriophage. And we can harness these powerful mechanisms therapeutically for gene editing, or in the case of RNA interference and so forth. Now, in higher organisms like vertebrates and humans, sequence-specific defenses like CRISPR-Cas and RNA interference can't keep pace with the evolution of the pathogens. And so you need a more general defense to detect foreign nucleic acids generally. And I discovered the first part of this puzzle in 1995 when I was in academia. And I discovered immune stimulatory CpG DNA. CpG is a cytosine phosphate guanine. And in our genomes, when the C is followed by a G, the C is generally methylated. In viral genomes and retroviral genomes, the C is not methylated. And the immune system evolved a particular receptor to detect that difference in DNA between our DNA and foreign DNA, and to recognize that as a sign of danger. And so I formed a biotech company to develop that using a synthetic CpG DNA. We thought we were going to cure cancer and make a whole new generation of vaccines. And actually, the vaccine part of it worked out well. There are 2 approved vaccines now with that. But the cancer part of it did not work out. The early form of the DNA we had failed in cancer trials. Pfizer acquired the company for the vaccine platform, but then in 2011, Pfizer abandoned cancer immunotherapy because nothing was working. And I thought that was going to be the end of the story, and I did some other companies. But then in 2014, checkpoint inhibitors were approved, the PD-1 checkpoint inhibitors. And I thought, here's another opportunity to come back and try this cancer immunotherapy again, we had a newer, much more powerful form of the CpG DNA that was a better mimic. And that led to the company Checkmate that was acquired by Regeneron a couple of years ago. At Checkmate, we did have monotherapy activity in cancer. Now, it was only in 25% of patients with advanced melanoma, but these were patients who had failed checkpoint inhibitor therapy. So that was a big step forward. And then I thought after Regeneron acquired us that was going to be my retirement and that I was done. But I kept thinking about the immune system and how the immune system works and what were we missing with the DNA, because all along I was thinking in the back of my mind that this is a defense mechanism that we're trying to harness here. And I started thinking more about what is it that we're really trying to mimic? What is it that we're trying to trick the immune system into thinking is going on? And I realized that really what we want to do is to trick the immune system into thinking that there is a retroviral infection in our own cells, because a retrovirus copies its genome into our cells. And once you have a retrovirus in your own genome, the only way you're going to get rid of that infected cell is by killing it. And the immune system evolved a whole class of cells to do that, called CD8 positive T cells. These are the basis for CAR T therapies. You can grow up boatloads of them and give them to cancer patients and treat cancer that way. But what if we could induce this in vivo? And I realized that we only had part of the puzzle at Checkmate because we were doing the DNA part of it. But after my early formation of my first company, receptors were discovered that detected viral RNA also. In particular, there's two receptors, toll-like receptor 7 and toll-like receptor 8. They can detect viral or retroviral RNA and distinguish it from self-RNA. And people have pursued that. That mechanism is also involved in mRNA vaccines, for example. But no one had ever put the RNA part and the DNA part together. And that combination of a viral RNA mimic and a double-stranded DNA, that's what you get in the replicative complex of a retrovirus that is about to integrate or has integrated into our genome. And this is really teleological deduction, but I hypothesized that structure, this RNA DNA structure, ought to be the strongest danger signal, if you will, that the immune system could possibly detect as a sign of this infection. And so I had a contract lab synthesize some new designs of RNA DNA hybrids that I thought would mimic this. And one of those was just extraordinarily active. It was more than 10 times more potent than the strongest activator that we'd ever seen before. Plus, it activates additional cell types that we had never co-stimulated with our previous compounds. And I just thought, I have to start a new company and develop
Couldn't
this.
help yourself.
What's going to happen with these? So I got together a bunch of folks that I'd worked with in the past, some of the Checkmate gang and some others. And we scaled up our lead compound. We're doing LNP formulation development right now. We're going to be taking it into mice and monkeys in the next few months. And our goal is to get this into humans next year. I'm not sure how we're going to fund it all, but at least we can fund it up through the monkey studies.
Yeah. So you wrote out a lengthy LinkedIn piece, which we'll put in the show notes, a link to it, describing how the plan anyway is to get to a point where you can out-license partner out the first medicine, the first candidate, and then use that to kind of keep self-funding. I want to come back to the science because there's so much cool stuff there, but since we're here now, it is your desire to kind of do this more bootstrapped based on just sort of the realities of the venture market, and so based on your 20, 30 years of building companies in the space and wanting to do things differently or have more control or whatever?
I would say both. I mean, I think in retrospect, we made a lot of mistakes in the past in the way that we were developing our technology. In my first company, Coley, for example, we had one lead CpG DNA. It's the first compound we took into the clinic. And for convenience, really, we licensed that same compound to multiple different pharmas, Pfizer, GSK, Novartis, Merck. They all had vaccine programs using the same DNA. And we could have easily had a different CPG for every different pharma, but it just seemed like the simplest way to go. Well, of course, what happened once Pfizer bought us is that the other companies all terminated their development of the CPGs that they were working on. And so one of the differences for Zola is I don't want to be beholden to a single pharma. I want to have different compounds for multiple different pharmas. I think this is a broad enabling technology that every pharma that is working in cancer immunotherapy is going to want to have one of these compounds. If we're right, these will be the strongest inducers of CD8 positive T cells of anything out there. In terms of the investors, investors typically want to take control of a company, and the direction that they want to go is not always the direction that I want to go. It may be the most lucrative to make a single big pharma deal. If I want to make multiple different deals, and if I want to have a compound for academics to develop as well, I don't want to have different goals of my investors. And so I've decided to find a way to finance this, that if we do have VC investors, they don't take a majority control. I want to be able to determine the direction of the company.
So given that at least for now it's kind of being bootstrapped, I'm guessing you're a pretty lean team. What does the team look like today?
So we have 14 people on the team, but most of them have day jobs. It's part-time. No one has a salary. People are simply getting paid in equity. The understanding is that once we do make a deal where we have some cash, then people will start getting salaries. But in the meantime, because we're such a small team and because everyone knows what they have to do, it's actually not a 40-hour-a-week job. Maybe for me, it's probably closer to 60 hours a week.
Sure.
But, we can do this. We're working through CROs. Each of the CROs has their defined mission. And I think we can get this to the point of even first in human, in fact, with a virtual team without necessarily having our own labs.
And where do you see the sort of, kind of first indications to try this in? Would you go back to melanoma, given the sort of accessibility and the prevalence of checkpoint inhibitors there, or would you start somewhere else?
So I've talked to our melanoma docs who were very excited about this new approach. In order to do first in human in melanoma, they want to have a dose before doing the first in human in early stage melanoma. And so to define that dose, I think we'll go into a different tumor type initially. And, I guess I'm kind of a risk taker here. I believe that this is going to be so powerful that we should be able to get responses in some of the greatest unmet medical needs for immunotherapy of cancer, including tumors such as pancreatic cancer, microsatellite stable colorectal cancer. If this theory is correct, we should be able to induce immune responses in those tumor types. And if we can't, we'll do translational studies to determine why not. Where is the blockade here? So I've actually been meeting with a number of investigators who would be willing to do first-in-human studies with us in tumors like pancreatic cancer, where we deliver the drug.
Some colder tumors.
Colder tumors, that's right. The classic colder tumors. If we're right, we should be able to overcome the immune suppression in those tumors because of the way that we're activating these immune cells. And we should be able to do it with an IV infusion using liposome or lipid nanoparticle delivery vehicles that are optimized for uptake into immune cells. The liver has a lot of immune cells, and we know from past studies that it's easy to deliver into those cells in the liver. In fact, it's harder to avoid them.
That was my next question, is how you intend to deliver this. So maybe you could talk about that a little bit more. And then once these nanoparticles and liposomes get into the liver and target immune cells, which are the immune cells in the liver that are uptaking and what happens from there?
Yeah. That's the key thing. So the key cell type for us is dendritic cells. Dendritic cells have been likened to the conductor of a symphony orchestra, or to use a more militaristic metaphor, the generals of an army. They are the commanders. They give directions to the other immune cells, like the T cells that I mentioned, that it's the T cells that are the soldiers of this army. They are licensed to kill invading cells. And in cancer, we're trying to activate the T cells to kill tumors. You can't do that using endogenous mechanisms without stimulating dendritic cells. And almost all cancer companies that are trying to develop immune activators to treat cancer, they're focused on activating dendritic cells. Where it's differentiated and what's unique about Zola's approach is the type of dendritic cell that we're focusing on. And without going into all of the complexities, there's at least half a dozen different subsets of dendritic cells. And in mice, essentially all of them can induce T cells to kill tumors. Everything works. And so if you're optimizing your therapies in mouse models, which is what most people do, you actually end up selecting the wrong type of dendritic cell to stimulate. And I think this is because of my clinical training. Before I went into research, I trained as a rheumatologist, and I learned very early on about the types of interferons that are involved with activating T cells in autoimmune diseases in humans. And this is particular a type of interferon called interferon alpha. And this is only produced by one extraordinarily rare subset of dendritic cell called a P1 plasmacytoid dendritic cell. Most people working in cancer immunotherapy don't even know about those. I'm shocked. I routinely talk to people at companies who are leading their cancer immunotherapy programs, and they've never heard of this type of dendritic cell. We know how to activate it. This retroviral mimic is what activates that type of dendritic cell to command the CD8 T cells to go and attack the tumor. So I think we're so far ahead of the other companies in our space. And to be clear, there are multiple other companies developing TLR agonists. There are companies developing TLR7, TLR8, and TLR9 agonists. None of them have put it together, and the molecular structure of the other agonists are severely defective, in my opinion. I'll give you an example. TLR7 and TLR8 can be activated with small molecule agonists, and they're a heck of a lot easier to develop than an RNA agonist because you can put them on an antibody, you can easily deliver these small molecules. What people ignore is that there are two binding sites on the TLR7 and TLR8 that they're trying to stimulate with these small molecule agonists. And the small molecules only bind one of those two sites. And people think it doesn't matter because they can measure downstream cytokine production, but they're measuring the wrong cytokines. They're not looking at the interferon alpha, which I have concluded based on my background as a rheumatologist is what you really want to induce. And the RNA does that logs better than the small molecules do. The small molecules actually drive inflammatory cytokines that have given TLR8 especially a bad reputation for toxicity.
I was about to ask, do you think you can avoid cytokine storm with this?
Absolutely. Well, maybe not avoid it entirely, but we should have way less of it than the small molecule agonists because we have a biologically different response. And so maybe it's good for us that other people in the field aren't aware of this, but it's been an educational process for me going and talking to potential investors because I have to simultaneously explain probably 4 new things to them that they've never heard before. And most people just have trouble keeping up with that. So we're just going to move forward. I think we have pathways to raise the funding we need to get the first in human data. And if I'm right, those data are going to be getting a lot of people more excited than anything I say will.
I hope you're enjoying this episode of Talking Precision Medicine, powered by Genialis. We'll return shortly with more of this insightful conversation. In this episode, Bart Krieg demonstrates how a deep understanding of how the immune system works inspired his latest therapeutic innovation. To learn more about how Genialis puts biology first in its approach to innovating the precision medicine space, you'll want to catch Genialis on stage at several events this fall. You can meet us in our hometown, Boston, at the World Clinical Biomarkers and Companion Diagnostics Summit, September 4 to 6, at the Pancreatic Cancer Action Network's annual meeting, September 14 to 15, and the RAS Targeted Drug Development Summit on September 25th to 26th. At this latter event, I'll be presenting a scientific update on Genialis KRAS ID, our first-in-class biomarker that predicts response and benefit of KRAS inhibitors. To learn more and schedule a meeting, please click the link in the description. If you enjoy this show and would like to hear more episodes with more great guests more often, there's one thing you can do to help. Subscribe. Please follow us on your favorite podcast streaming service and leave us a review or comments. Thanks, and now back to the conversation. I'd like to zoom out a little bit. When you were describing the nature of the treatment, so you've got your double-stranded DNA and it's hybridized with a single-stranded RNA. I think a lot of listeners who aren't familiar with this kind of therapy might recognize that as starting to sound a little bit like the flavors of COVID vaccine that came out, right, and really changed the game. But also for scientists who listen, it sounds a bit like how you do CRISPR, right? And I guess that's kind of the point. Talk to us a little bit about the evolution of oligonucleotide therapy, of nucleic acid therapy. This has been a big part of your career. How has the convergence of all these other nucleic acid-based medicines kind of set a stage for you now?
Let me just quickly recap the nucleic acid therapeutics field, because it was when I was a postdoc at NIH training as a rheumatologist that I first got excited about this. This is back in the 1980s that people first were proposing to develop antisense oligonucleotides as a therapy. And I started using those as a tool at NIH. There were a whole bunch of people getting excited about that. The first companies got formed. What is now Sarepta was formed. What's now Ionis was formed. Several other companies that have since gone defunct, because it turned out that the early chemistries and designs that people were trying to develop just didn't really work. And so during the 1990s, there were repeated setbacks, although there was one oligo that got approved. It was a commercial flop. And then in the 2000s, people got all excited about RNA interference because they thought that might be an easier pathway forward. And there were lots of exciting data. Alnylam got formed in 2002. But then during the 2000s and even into the early 2010s, there were still repeated setbacks and failures, despite investment of billions of dollars. In the late 1990s is when I discovered the immune stimulatory DNA. We had our first human vaccine data in 1999, and we started partnering with companies. It took until 2017 for the first CpG vaccine to be approved. And that was approved based on work of one of our competitors, Dynavax, because the companies that we licensed the technology to never developed it. For various reasons. So it was in 2016 that the first successful antisense oligo was approved, nusinersen. 2018, we had the first siRNA approved. Since then, there have been multiple other siRNAs and antisense oligos, even another CpG vaccine. So the field of nucleic acid therapeutics today, we can count at least 24 approved nucleic acid therapeutics, including mRNA vaccines. So coming back to your question, what has enabled this field has been an explosion of work on nucleic acid chemistry. We have building blocks now to design all kinds of different nucleic acid functions and tune in or out various properties. So for my own work now at Zola, building on all of that experience, it's relatively easy for us to go out to contract manufacturers and have them make whatever it is that we want. And then with all of the work on formulation development, we can build on that to deliver these compounds into patients with a high confidence of getting into the targets that we want.
Tell me a little more about the molecule design piece of this. Is it just ration design? Are people using AI-based tools? Is it instinct? Is it trial and error? Like, how do you approach figuring the actual sort of compound optimization?
Yeah. So first of all, for the field in general, for siRNA, antisense oligo, RNA editing, AI and ML are playing a big role in the work of various companies in terms of processing large data sets because of all of these different modifications, that it's very site dependent where you put the modifications and how it affects the properties of the oligo when you're basing this on hybridization to targets inside the cell. Modifications can increase or decrease affinity at specific locations, and that's important for off-target effects. Because with a hybridization-based molecule like this, you have your intended target, but you want to tune it so that you're not interacting with unintended targets. And you mentioned CRISPR-Cas earlier, designing these compounds to be effective with the incorporation of modifications. That requires large data sets typically in processing. For us with immune stimulation, we're optimizing this based on sequences that have been recognized on crystal structures of the receptors. So we have crystal structures for these toll-like receptors. We know exactly which bases fit into each of the different sites. And so then it's a simple matter to design a nucleic acid that contains those sites. Now, there's some other tricks to it too, of course, because we want these to assemble into a structure that looks like the replicative form of a retrovirus. And we think we know how to do that. So it's...
That's arguably an easier problem in protein folding, but...
I think it is, yeah.
4 bases instead of 20, 20 amino acids.
We don't have to make thousands of oligos. To come up with our lead compound, the lead compound is number 7 of the oligos that I make for Zola.
No, it's so fascinating. Maybe you could go into the potential of the sort of platform play here a little bit. So, we've talked a bit about where you want to go first, but, you've described a business model where you kind of have one asset per partner so that the IP is clear and things get developed. But, what are some of the other uses? Seems like there's even something to do with cell therapy, possibly with, checkpoint inhibitor proteins. Where do you see, the kind of interesting opportunities that maybe aren't obvious to others?
Yeah, no, that's a great question. So there's 2 aspects to this. For our lead program is designed to co-stimulate 3 immune receptors, toll-like receptor 7, toll-like receptor 8, and toll-like receptor 9, with a single compound delivered intravenously with a lipid nanoparticle. That could be used in combination with just about any immune therapy, checkpoint inhibitors, activators of myeloid cells, CAR T therapies. It could also be added into vaccines. So the issue with vaccines, you need 2 components for a vaccine. You need an antigen, and that gives the T cell the direction for what it's supposed to kill. And you need what's called an adjuvant, and that activates the dendritic cells to give the T cell the license to kill. The adjuvant part has been the hard part with cancer vaccines because all adjuvants work in mice to license those T cells, and then they typically fail in humans. We think that our adjuvant is going to be the best for licensing these T cells in humans, and we think it's going to replace others. So that's the first part. We can use this very broadly across many different applications. But I mentioned before, we want to work with multiple different pharmas. And so our approach to develop this platform is our lead compound will be licensed exclusively to a single pharma or single partner for them to develop in whatever combinations they want and whatever tumor indications they want, very, very broadly. We're developing other new compounds that have other properties and may hit those same receptors or may add in other receptors. Some companies have told us they think TLR8 activation is going to be too toxic, so we've designed compounds that will just activate TLR7 and TLR9. And in some patients, I think that will be adequate.
Well, that's an amazing rheostat to be able to do that.
Yeah, it really is. It's a rheostat. And the other part of it is we want to be able to put these into other delivery systems. Some companies want to put them on antibodies or other targeting. For that, we have to incorporate modifications into the RNA and DNA to stabilize them against serum nucleases. Because right now we're using mostly native backbones. These are not serum stable. So we'll be having additional generations of compounds that we can license with other partners also exclusively, or that's our dream.
Well, I think, the timing of it is it could, in my opinion, could be better between the flurry of cell therapies that I think would benefit, but also things like, with pembrolizumab going off patent, this could be a really, really powerful way for whatever the successor of that drug is to work better than, what's coming.
Yeah. The future of cancer immunotherapy is in combinations. In the early clinical development of the precursor compound to what we have now at Checkmate, this is called vidutolimod, is the name of the drug. And that's been in hundreds of humans with advanced cancer. What we learned with that compound is that in patients with advanced melanoma whose tumors had failed to respond to other drugs, we saw monotherapy responses with vidutolimod injected into tumors. And those responses were systemic. Tumors shrank elsewhere as well. But they were brief. As a monotherapy, the duration of response was less than 6 months. With the frequency of response was 9 out of 40 patients, so a little over 20%. When we combined this with a checkpoint inhibitor, the checkpoint inhibitor improves the duration of the response from less than 6 months to more than 2 years, because we think we're inducing these T cells and the checkpoint inhibitor keeps them from being turned off. The frequency of response was similar in the combination arm, but they were systemic responses. So the future of cancer immunotherapy is in combinations. And with Zola, we'll do initial development as a monotherapy, the way you have to in early trials, but with the anticipation that this will be added in with other drugs.
No, it sounds like, given that you've been at this sort of, developing this class of medicine since 95 and have done a few companies, you've learned a whole lot of lessons that you can sort of build off of.
Some of the lessons were painful. When our first phase 3 trials failed with Pfizer, it was like a kick in the gut. I really thought it was going to work with that initial structure that we used. And since then, other companies have also had phase 3 failures with similar designs of oligonucleotides that really didn't mimic the structure of a retroviral genome very well. I think one of the lessons from this in terms of what has worked and what has not worked is that the more closely you mimic the structure of a retroviral genome, the better the results have been.
I think a lot of people don't appreciate kind of the nonlinear path that most things that are today medicines have had to take to get to become medicines. It takes a long time, and part of that long time is that things fail, things get put down, things get picked up. Tell us just kind of one illustrative story of sort of one of these windy paths from your career so far. I mean, vidutolimod might be it. I recall that was one of these that got put down and picked up a couple times.
Yeah, it did. Absolutely. So the story of vidutolimod was this was a drug that was originally being developed by a Swiss company called Cytos. And they were hoping to get around my patents at my first company, Coley, by using a different type of CpG structure, CpG DNA structure, and packaging it in a virus-like particle. And they were developing this for vaccine applications, but then focused on allergic immunotherapy of patients with allergic rhinitis and asthma. And they had some early positive data in patients with mild allergic rhinitis and asthma. But the key market there is in more severe disease. And by the time they started their phase 2 clinical trials, Coley had already long since been acquired by Pfizer, and Pfizer had abandoned the entire field. So I joined the Cytos board. I think that was in 2011 when they were trying to develop this for asthma immunotherapy. The phase 2 trials failed there, and the company was possibly heading into bankruptcy. By this time, it was about 2013, and actually, 2013. And I, in early 2014, I joined Sarepta as Chief Scientific Officer. I was really excited about their technology for treating Duchenne muscular dystrophy with exon-skipping oligos, and I didn't think I'd ever be able to come back to CpG again. But it turned out I got fired from Sarepta. I was actually escorted out of the building by security, and that was a traumatic event for me that had the good outcome. I went out backpacking to kind of clear my head and think about things. And I realized that the drug that Cytos was developing that by now had failed in asthma could be repurposed for cancer immunotherapy. And that I had an opportunity, because now the first PD-1 checkpoint inhibitors had been approved. I had an opportunity to combine this drug that had already been in humans with a checkpoint inhibitor. And I remember lying in my tent at night thinking about this and thinking about different designs of other CpGs we could make. So anyway, I ended up licensing the drug from the board at Cytos, with whom I had a friendly relationship, and had friendly investors to help me found Checkmate and repurpose that drug for cancer immunotherapy. And almost immediately, I think the fourth patient in our clinical trial, and this was just during the dose escalation phase, the first patient at the 3 milligram dose had a remarkable clinical response, and we knew we were onto something.
That's a great story. I'm glad you mentioned backpacking and kind of sleeping and pitching the tent. Most folks who know Art know that he likes to spend a lot of time in the mountains, and if he's not in the mountains, he's running along the Charles or the trail somewhere. Talk to a little bit about that part of your life and how you feel like it plays into this kind of energy you have for innovation and company formation. I mean, it's hard to build companies, right? It's not just doing science. Like it's an uphill climb.
Yeah, it is so hard. And I think that for me, I believe that a lot of the decision-making process and things, it comes to me after a long, drawn-out thought process that is largely subconscious. It's hard to, some decisions you make consciously, but the broad strategic ideas are more from the subconscious mind. And I find that going out trail running for a day or a night, you're totally focused on your foot placement and everything else. You're occupying that part of your mind, and your subconscious can work on these other issues while you're on the trail. And I come back from it, and often, not always, I find a solution to something that I've been thinking about. Now, I will add, and I don't know if you know this, last fall in August, I was backpacking in the Sierras, going cross country with some friends. And I had my first ever serious accident, descending a steep ski slope with a backpack. Rock rolled, and I fell, and my left knee got pinned under me in a way that it was hyperflexed, and I had ruptured my quadriceps tendon off the patella. You can't walk with that. And we were 2 days away from any cell service. But fortunately, I had a Garmin InReach with me, which is a satellite device that you can call for help. And within a few hours, I was being whisked away by a helicopter to a trauma center and had surgery to repair it. That was September 5. And then, the recovery process from something like that is so frustrating. When you like to be active like me, the surgeon told me it would be 6 months until I could run again. And I remember telling him, oh, well, that's okay, because that means I can run the Boston Marathon, because this was September 5 and Boston Marathon was April 15. And he looked at me like I was joking. And then he realized I actually wasn't joking. And he said, he did the mental math. He said, you'll only have 6 weeks to train, you realize. And I said, yeah, but I want to give it a try. Do you think I can? And he said, well, let's see how it goes. And he gave me clearance to try that as I followed up with him. And I'm happy to say a few weeks ago, I finished the Boston Marathon and
Amazing.
Was able to even get a qualifying time to do it again next year. So it was my 9th consecutive one. My goal is to get at least 10. So I like stretch goals like that.
That's really astonishing. We had a few guys from our team. They hadn't qualified for the full Boston Marathon, so they decided to do something pretty harebrained, which is to run the route, but in reverse at night, the night before. They had this assumption that the route would be closed because, it was like late at night the night before. Surely the route's closed, right? Now, why in reverse, I'm not entirely certain, except I guess that way you're coming back in the city, so it's easier to get home. But, the Boston Marathon is slightly downhill, and so running it in reverse is uphill.
Uphill, yeah.
But as it turns out, only the first couple of 100 yards from the start were actually closed. So they were running through traffic, dodging buses. It turned into one of those, there's the Patagonia fun scale, type 1, 2, and 3, and they describe this as like type 5 fun.
Yeah, that is tough. Yeah.
Tell us a little bit about the Timmerman Traverse hike you go on, and maybe we can, some listeners might want to get involved or might want to do sponsorships or something like that.
Yeah, that'd be fantastic. Luke Timmerman came up with this idea to raise funds for various charities. Life Science Cares is one of them, by getting together a group of biotech executives who are not necessarily hikers, but are reasonably fit, and challenging them to do something outdoors. He's done the climb of Kilimanjaro, hike to Everest Base Camp. and the one that I've helped Luke on for the last 3 years is the Timmerman Traverse, where we do a 3-day hike across the Presidential Range in New England, covering 8 of the tallest mountains in the Northeast. And it's just been so much fun for me. I mean, I've done this traverse in the day and at night and winter, but doing it with people who are novices sometimes, and helping them to do this safely and to have fun along the way and an experience. I have just been so energized, and my life has been enriched by that. So I really enjoy that. And it's raised a lot of money, millions of dollars for charity. We do seek corporate donors, and anyone who might be interested in becoming a corporate supporter of the Timmerman Traverse can certainly reach out to me, or just Google Luke Timmerman Traverse, and you'll find links to support us.
Awesome. We'll stick some of those links into the show notes. All right, this has been great. Just parting thought, given what, you're innovating now, if you get it right, if this works the way you think it will, what does the cancer therapeutic landscape look like in 5 and 10 years?
Yeah, I think the cancer immunotherapeutic landscape will involve much less traditional chemotherapy. And I think that CAR T therapies will be replaced by these CD8 T cell inducers. Instead of growing up large numbers of CD8 T cells outside the body and giving them back to patients, we will be able to induce them endogenously using a combination of this retroviral mimic that triggers these 3 pathways that evolved to induce this cell type. We'll be using that in combination with checkpoint inhibitors and other therapies. I think essentially every cancer patient will be treated with a cocktail of 1 or more checkpoint inhibitors, an innate immune activator, which will be one of these TLR789 agonists, and maybe 1 or 2 other compounds. Precision medicine for oncology, I think, may be a supplement to it. But, the precision medicine approaches typically give short-lived responses. In order to get a long-lived response, you need immunotherapy. So it may be used in conjunction with precision medicine. Maybe you induce an initial response with a precision medicine therapy, but then you solidify that with an immune therapy that is able to evolve with the tumor, potentially, if you design this in the right way, to achieve that long-lasting response.
Amazing. Good luck with everything. Honestly, we're all rooting for any patients need this, and I think it's just such a cool culmination of science and technology and experience, and a great story.
Well, thanks. One other thing I forgot to mention earlier is where the name Zola Therapeutics came from.
Yeah, tell us.
My first company, Coley, was named after the surgeon, William B. Coley, who in the 1890s injected patients with cancer with bacteria to try to stimulate an immune. Well, he thought that there were toxins in there that would kill the tumors. We now think of him as the father of cancer immunotherapy, and that this actually stimulated responses in some patients. The name of the first patient that he treated was Zola. Now, we don't know if it's a first name or a last name. The medical records simply say Zola. But for me, it's coming full circle from my first company, Coley, honoring the surgeon, to my last company, Zola, honoring the patients. Because as you said, it's all about the patient. And our whole business model, our whole technology is aimed to bring this to as many patients as we can.
Fantastic. Thanks again.
Thank you. Take care.
This has been another episode of the Talking Precision Medicine podcast, brought to you by Genialis. Hopefully by now you've subscribed to the show, but if not, go ahead and click that subscribe button and leave us a review. And please do pay us a visit at www.genialis.com. That's www.genialis.com to learn more about how AI is transforming precision medicine. Thanks for listening, and see you next time.